For joining today. My name is Hugh Davis. I'm head of the commercial team here at Alstone. Before handing over to Giuseppe to go through some of the results of our liver biomarker program, I just wanted to share a few details on Alstone as a business and some of the technologies that we've developed for breath analysis. So our mission as a company is to save 100,000 lives and 1.5 billion in healthcare costs, and we'll achieve that by being the global leader in breath analysis for early detection and precision medicine. On the early disease detection side of things, we obviously have our in-house development programs in areas such as oncology, liver disease that you're going to hear about today, exposure, as well as certain infectious diseases. We also work with a wide range of academic partners, both in Europe and the US, across a wide range of different disease areas and clinical questions, deploying our breath biopsy technology for non-invasive biomarker discovery and validation. On the precision medicine side of things, we tend to work with biopharma companies, large pharmaceutical companies such as GSK and AstraZeneca. And there again, we work across a range of different disease areas. To date, we've conducted a fair amount of work in the respiratory disease areas, looking at diseases such as asthma and COPD, looking to stratify patients for optimal therapeutic response or to identify and deploy biomarkers for drug efficacy and safety, for example. So what are the types of biomarkers that we can potentially measure on breath? They fall into two main categories, those that are volatile and those that are non-volatile. Our core business has been around the measurement of volatile organic compounds, and these can be both endogenous metabolites that can originate from biology that's ongoing both within the airways as well as other organs and tissues in the body. But they can also be exogenous compounds that can come from the environment and the air that we breathe. And this could be dietary components, they can be pollutants and other molecules in the air. And we can measure all of these molecules on breath. In addition to that, we're also developing out a new capability around the measurement of respiratory droplets or breath aerosols. And these are larger macromolecules that are more water-soluble, molecules such as proteins that we can measure via immunoassay, for example, viral DNA, RNA, viral particles, obviously highly relevant to the current situation, drugs as well as non-volatile metabolites. And we're working on the development of this receiver breath sampler that allows us to measure both the VOCs, an area that we have a lot of experience, as well as capturing these respiratory droplets on a filter within the device and measuring them from the same breath sample. So we provide both breath biopsy services through our labs in Cambridge in the UK. We also provide various technologies that we can supply as products and place in customers' labs where they would like to do the analysis themselves. Our core technology is the receiver breath sampler and breath biopsy kits that accompany that. This really allows for a very reliable in vivo breath sample collection of both volatile organic compounds as well as respiratory droplets for subsequent analysis. In terms of the analysis itself, we can, as mentioned, look at both VOCs as well as larger macromolecules. On the VOC side of things, we can do a broad-based, comprehensive VOC analysis with our breath biopsy Omni platform, and also look at selected VOC panels. And we've recently launched a respiratory panel to look at respiratory inflammation as one example. We can also perform in vitro work as well. So we have the capability now to look at the head space, so the air above tissues, cell cultures, and isolated macromolecules to measure what VOCs are being emitted from those types of samples. And that could be then hypothesis generating and allow for the development of a targeted panel, which then can be potentially measured on breath. On the product side of things, we can place this breath biopsy collection station, which I'll talk about more in a moment, along with the breath sampler in the facilities of people that own a thermal desorption GCMS technology. We also have our own Lone Star VOC analyzer, which is based on field asymmetric ion mobility spectroscopy, or FAMES technology, which is a small footprint analyzer for VOC analysis, again, which can be done in your own lab. So I just want to talk through a little bit more about the components of the breath biopsy collection station. And core to this is the receiver breath sampler that we've developed and commercialized, which allows you to collect a very reliable and reproducible breath sampler that's hooked up to a laptop, which controls the device, measures the normal tidal breathing, and then activates pumps to draw the volatiles from the breath down onto sorbent materials, which we call the breath biopsy cartridge, which are the green tubes that you can see here on this particular device. Those tubes are then uncoupled from the receiver collector and sent back to our labs, where we do the downstream mass spec analysis. Also core to this setup is the Casper portable air supply. So that Casper device plugs into the receiver breath collector and basically scrubs the background VOCs from the ambient air that's in the room where the sample is being collected. So that's providing a clean air supply, which vastly decreases the background contamination that can come from VOCs in the environment, and thus boosts the signal to noise. So those are the components of that station. We supply that within clinical studies, clinical trials for groups that are doing service work with us, but we can also sell this as a collection station to groups that have the capability to do their own analysis. I just want to talk a little bit more about the analysis that we can do on the back end through our labs in Cambridge, through this breath biopsy Omni assay setup that we now have, which is incorporating high resolution accurate mass GCMS capability that we've recently installed. Also core to this is a library of VOCs that we're building over time, which allows us to accurately identify and tie into the biology VOCs that we measure on breath. So it gives us more of a mechanistic idea about how VOCs are being produced and how they relate to the biology. So just to talk a little bit through this workflow, the TD samples, so the breath biopsy cartridge that's collected from that receiver and then shipped to us, to our labs in Cambridge. Once that arrives, we then, it's barcoded so we can log and track it through the system. We then can perform a thermal desorption GC mass spec analysis using high resolution Orbitrap mass spectrometry. And within this device, we can do, within the system, we can do either untargeted VOC discovery type of analysis, where we can look at a wide breadth of different VOCs in parallel, extract that data, match it against the library, and then output a list of VOCs that are showing some sort of fold change in response to disease or drug treatment, for example. In parallel or separately, we can also do a targeted VOC analysis, which is more hypothesis-driven. As mentioned earlier, we've recently developed a respiratory inflammatory type panel of VOCs that can be measured in more of a targeted fashion. And the beauty about the system is that we can do both the targeted analysis as well as untargeted to look for new things within the same analysis. I'm now going to hand over to Giuseppe, who will go through one of the case studies that we've got in the liver area based on our in-house test program.
Hello, my name is Giuseppe and I am a metabolomic scientist at Allston Medical. Today I'm going to show you some interesting results about breath limonene, which is a potential biomarker for the diagnosis of liver cirrhosis. Our well-being depends also on the health of our liver. And unfortunately, there are different factors that can affect liver function. One of these is the excessive accumulation of fat in the liver without excessive consumption of alcohol. This condition is called non-alcoholic fatty liver disease. It's a benign condition, often asymptomatic, which can progress to non-alcoholic steatohepatitis, a disease in which inflammation of the liver takes place. The constant injury of the liver leads to accumulation of fibrotic tissue, which results in liver cirrhosis. As long as the liver is able to perform the physiologic tasks, we talk about compensated cirrhosis. However, when that's not the case anymore, episodes of decompensation may occur. And liver cirrhosis can lead to different complications. One of these is hepatocellular carcinoma, which is the most common type of liver cancer. We may have liver failure, in which the liver is not able to perform the physiologic tasks anymore, or hepatic encephalopathy, a condition in which cognitive functions are affected because of excessive accumulation of ammonia in the liver, which is not cleared up efficiently anymore. Therefore, detection of liver diseases at the stage of non-alcoholic fatty liver disease, or NASH, is crucial for early intervention to prevent future complications. And it's worth also to mention that the whole process from an healthy liver to cirrhosis may take years. The number of people affected by non-alcoholic fatty liver disease is increasing around the world. For example, if we consider the United States and some of the main European countries, we can see that more than 100 million of people are predicted to be affected by non-alcoholic fatty liver disease. And of course, this is going to have massive impact on the healthcare systems of those countries. Given that this condition involves so many people, what is the state of the art for the diagnosis of non-alcoholic fatty liver disease? In general, liver diseases can be detected through blood biomarkers, for example, alanine aminotransferase. However, when these biomarkers are altered, further investigations are necessary. The gold standard method used to diagnose non-alcoholic fatty liver disease is liver tissue biopsy. However, this is an invasive procedure and can lead to complications. Other less invasive techniques are the ultrasound and CT scans or transient elastography. However, all of these conditions have one weak point, which is lack of measurement of liver function. Indeed, we are going to measure other parameters, but we are not having a readout of the functional capacity of the liver. It is essential to develop specific functional tests able to determine if therapeutic interventions improve the liver function. Our goal at Allston Medical is to develop a functional breath test able to detect non-alcoholic fatty liver disease, or NASH, in a non-invasive way. Where to start? So we started from the literature, and we found a handful of studies where the authors compared the breath of patients with cirrhosis with that of healthy volunteers. They found different biomarkers, but one of them was consistent among all the studies, and this biomarker is limonene. So, among all these studies, there is one of particular interest for us from Professor Chris Mayhew, University of Birmingham, in which they measure the levels of breath limonene before and after liver transplant in patients affected by cirrhosis. As we can see, before liver transplant, patients with cirrhosis have significantly elevated levels of limonene compared to controls. However, after liver transplant, levels of limonene in the breath normalize in the following weeks. Another interesting follow-up study from the same research group compared the levels of breath limonene in patients with cirrhosis complicated by hepatocellular carcinoma and patients with cirrhosis alone. As we can see, patients with cirrhosis have remarkably high levels of limonene in the breath. However, patients with hepatocellular carcinoma have moderately elevated levels of limonene in the breath. The authors were not able to explain this result because it may be due to the development of hepatocellular carcinoma or to an earlier stage of cirrhosis in the ACC group. We answered this question by matching the stage of cirrhosis severity between the two groups, and I'm going to show you the results later. Why limonene increase in the breath of patients with cirrhosis? According to the current model, limonene is a flavoring agent widely used in food and beverages. Therefore, we ingest high amount of this compound with our diet. In the liver, limonene is metabolized by two CYP enzymes, CYP2C9 and CYP2C19, and the bioproducts are excreted in the urines. However, in a cirrhosis condition, CYP2C9 and CYP2C19 activity is reduced, therefore biotransformation of limonene is also reduced. That leads to accumulation of this compound in the adipose tissue, increasing the permanence in the body and resulting in increased secretion in the breath. So, we designed a pilot study with the goals to have a proof of principle that a breath biomarker could be used to detect cirrhosis or hepatocellular carcinoma. We wanted an independent validation of the literature results, measure the performance and correlation with blood biomarkers, and identify possible confounding factors. Here are some inclusion criteria like age, gender, alcohol consumption, and there is one that I was mentioning before, which is the severity of liver cirrhosis measured as Child-Pugh class. And we matched the class between patients with only cirrhosis and patients with cirrhosis complicated by hepatocellular carcinoma. So the study has been performed at the Addenbrooke's Hospital by collecting breath biopsy samples with the Resiva system. So at the hospital, other information about patients have been collected, and this information, as well as sorbent tubes containing the chemicals from the breath, have been shipped to Alston Medical and analyzed by gas chromatography mass spectrometry to measure the amount of limonene in the breath for following analysis. And what are the results of our work? So, as we can see, patients with cirrhosis have significantly elevated levels of limonene compared to healthy volunteers. And this results is the same if we look at the levels of limonene of patients with cirrhosis complicated by hepatocellular carcinoma. However, if we compare the two disease groups, we found no significant difference. We gathered patients with cirrhosis and patients with cirrhosis plus hepatocellular carcinoma in one single group for further analysis. So to estimate the accuracy of a potential limonene breath test, we built a ROC curve, and we found an area under the curve of 0.78 and the Uden index corresponding to 10.2 nanogram per liter of limonene in the breath as a threshold to discriminate between healthy and diseased population. And this graph, as I mentioned before, has been obtained by comparing limonene levels in the breath of healthy volunteer with that of patients with cirrhosis, regardless the presence of hepatocellular carcinoma. Then we investigated correlation of levels of breath limonene with the blood biomarkers. But which one to choose? So we chose bilirubin, which is a potential biomarker of reduced liver clearance when it is increased in the blood. Then we chose albumin and prothrombin time measured as INR, which are potential biomarker of reduced protein synthesis capacity of the liver. And then another biomarker, alanine aminotransferase, which is a biomarker of liver damage. Although bilirubin, albumin and INR may be used as a reference for liver function, these are quite unspecific because they may be altered also by other factors. As we can see, limonene positively correlates with bilirubin and INR and correlates negatively with albumin. However, interestingly, we found no correlation with alanine aminotransferase. Bilirubin, albumin, and INR are blood biomarkers used to calculate the Child-Pugh class. Here we can see the algorithm. So, based on the magnitude of the alteration of this biomarker, a specific score is assigned to the patient. And the sum of all the score determines the Child-Pugh class, and therefore the severity of the disease. Consistent with the results I showed you before, we found that patients with the Child-Pugh class B have significantly elevated levels of breath limonene compared to patients with Child-Pugh class A. Then we investigated possible confounding factors. One of these is exposure to products that contain limonene, such as orange juice. And this estimation have been performed by asking the patients how often they were drinking or eating these products. We found that patients exposed to limonene on a daily basis have significantly higher level of limonene in the breath compared to patients exposed with the less frequency. We also investigated association with drugs by asking the patients which drug they were taking the week before breath collection. We found that patients taking substrates of CYP2C9 and CYP2C19, the two enzymes I showed you before that metabolize limonene, have significantly elevated levels of limonene in the breath compared to patients not taking these drugs. Surprisingly, we found no differences for patients taking CYP2C9 and 19 inhibitors. This result may be explained by compensatory mechanisms taking place and involving other CYP enzymes. So, in conclusion, we found that limonene increases in the breath of patients with cirrhosis. Occurrence of hepatocellular carcinoma does not affect breath limonene levels. Exposure and drugs are confounding factors that influence the amount of limonene in the breath. Elevated breath limonene correlates with cirrhosis severity and blood biomarkers representing clearance and protein synthesis capacity of the liver. So from these results, we can see that limonene has a potential as biomarker for the diagnosis of liver diseases. But which one is the way forward? So we are looking to improve the performance of a limonene breath test by using a dynamic approach. And we've broken down this approach in different steps. So in a first step that we call phase 0, we establish the best administration strategy in healthy subjects, meaning that we aim to deliver orally a known amount of limonene and measure the washout in the breath in the following hours. Then we investigate if this approach is able to discriminate patients with cirrhosis versus healthy controls. And in the case we get a negative result, for example, an area under the curve lower than 0.9, then we can go back and optimize the dosing or identify other probes targeting different pathways. But in the case we have a positive readout, for example, a good performance measured as an area under the curve higher than 0.9, So then we will investigate the same test in patients with an earlier stage of liver disease, such as NASH versus healthy controls. Excel breath contains several compounds. The breath biopsy omni-assay allows simultaneous targeted and untargeted analysis on the same samples. So besides limonene absolute quantification, we can obtain a peak area for other metabolites called volatile organic compounds in an initial stage, or VOCs in short. These other VOCs can discriminate between cirrhosis and healthy subjects, and I'm going to show you some preliminary results about this approach. A volcano plot generated by using the new volatile organic compounds and showing the log2 fold change and the log10 p-value identified different VOCs that are upregulated in the breath of patients with cirrhosis. And we found that limonene is among those VOCs as expected. Some other VOCs are downregulated. On the right, we can see a table summarizing some statistics for the most interesting VOCs. So here we can see on the left a box plot showing limonene, which is upregulated as expected. And on the right, we are showing the box plot for other interesting VOCs that are also upregulated. The question is if combining different VOCs can improve discriminatory accuracy between patients with cirrhosis and healthy controls. To answer this question, we constructed a ROC plot by combining limonene with other interesting volatile organic compounds. And as we can see, the area under the curve that we measured is 0.83, which is higher than the 0.78 found with the limonene alone. A permutation test we can see on the right suggests that these results are not random, and it were to investigate them further. So in conclusion, we have seen that combining the different discriminatory VOCs together with limonene, we can improve classification. Our next step are to validate the identity of these new VOCs and investigate the molecular pathways involved in the metabolism of these compounds to have more mechanistic insights. With that, I would like to thank you for your attention and to acknowledge all the collaborators that contributed to this study.
So thank you, Giuseppe. And I just want to provide some summary comments before we close. Just talk through a little bit about the future directions of that liver program and also how you can potentially work together with Alstone. So one of the immediate actions here obviously is to complete the analysis of the untargeted results that Giuseppe's shared in combination with the targeted evoke strategy that he also mentioned, using a standardized dose of limonene as an evoke probe. We then want to extend the investigation beyond late-stage cirrhosis to earlier stages of liver disease such as NAFLD and NASH, where there's a high degree of interest within the field, as well as extend the analysis to other biochemical pathways where we can develop evoke probes that are complementary to the limonene method. And then by having a combination of evoke probes, the hope is that this will allow us to achieve high clinical performance in terms of specificity and sensitivity. Our end goal being the development of a clinical test for the detection and monitoring of NAFLD and NASH progression. In terms of how we work, I spoke about it earlier. We have the ability to run services with both academic and commercial partners running clinical trials, whether they be interventional or non-interventional. We're very interested in speaking with partners that have relevant samples in the liver disease area, such as NASH, that would like to work together with us on this liver program. But of course, we can work on a wide range of other disease areas as well to identify and validate non-invasive biomarkers on breath. Thank you.
Thank you everyone for taking the time to listen to us today. And once again, sorry for the technical difficulties at the beginning. We now come to the question and answer section of the webinar. We've got a few questions in already, so we're going to begin with one of those. But if you'd like to ask Giuseppe, Mark, or Hugh a question, then please do send a question in via the chat function on Zoom. So our first question is, what are the possible approaches to improve the diagnostic accuracy of a breath test? This is for Giuseppe.
We are currently working in optimizing the approach, and there are several ways, several paths that we are considering to follow. So one of these is to ensure exposure, because as we have learned from our study, it's an important factor determining the amount in breath. So if we ensure that all the patients and all the controls are exposed, then we could increase accuracy. Another approach that we are considering is to investigate other metabolic pathways. And that can be done by using other evox that are metabolized by different enzymes. And this is also another way that we are following. And of course, if you have any other idea and you will discuss more, I will be more than happy to discuss.
Thank you, Giuseppe. So we've got another question here. Among the different studies, why was limonene the only biomarker identified consistently?
Okay, so basically limonene is special, and that's because it's highly fat soluble. And yeah, actually I was having this discussion with Chris Mayo by email some time ago, and indeed in their paper they found also another biomarker increased, which was 2-pentanone in patients with cirrhosis, and then they didn't found it right after liver transplant. So it was already metabolized. And the difference is probably due to the fact that the limonene accumulates in the body when it is not metabolized. And this is the reason why it's the biomarker prevalently found in different studies. But of course, we can look specifically to other biomarkers and administering them right before breath collection, and we can look at the washout and therefore rely also on other biomarkers.
Thank you. Are there any other compounds you could use as an EVOC probe identified in untargeted analysis?
Okay, so I was very curious to see if there were other compounds interesting there. Of course, we are still at an initial stage, so we have to validate the identity of those compounds. But from a first screening, I identified around 40 molecules that are GRAS compounds metabolized by different pathways, and those have potential to be used as EVOC probes. But of course, I repeat, the identity needs to be validated, and we are working on that right now.
Okay, we've got another one just come in here. So, have you checked other drugs beside those involving CYP2C9 and CYP2C19 activity?
Sorry, can you repeat the question?
Sure. So, have you checked other drugs beside those involving CYP2C9 and CYP2C19 activity?
Yes. So, we looked in general also to other drugs, and we found no differences in limonene between patients that were taking those drugs and patients that were not taking those drugs. In particular, we looked at antibiotics and anti-acid drugs.
Okay, thank you. Another one here. Are you exploring other metabolic pathways that could be assessed with this approach?
Yes. Currently, we are exploring metabolic pathways because we are exploring mitochondrial pathways. So mitochondria are very abundant in the liver, and therefore we think so those are promising pathways that may give a good discriminatory capacity between healthy and people with cirrhosis.
Okay, thank you, Giuseppe. And we've got another one just come in, and it says, what are the advantages of using an EVOC probe compared to endogenous biomarkers?
Endogenous biomarkers
are useful because they give a result. Basically, we don't need to administer anything. So, but on the other hand, they cannot be administered. Of course, we cannot administer bilirubin to people or albumin or other biomarkers that are routinely used as endogenous biomarkers. So on the other hand, with the EVOC probes, we can administer the molecule and therefore challenge the pathway. And that gives a good, probably is going to potentiate the discrimination capacity. First, because we can measure it well above the background levels. And second, because we saturate the pathway and we can measure more fine alterations that may happen when there is a chronic liver condition.
Okay, so I think that's all the questions that we've got in. There does look like there's somebody that's trying to ask us some questions on the chat. So if you have got a question, then please do write one in. If not, I think we'll wrap it up there. So thank you very much, Giuseppe, and thank you very much, everyone, for joining us. And we hope to see you all at the Breath Biopsy Conference. The details are on this slide here. We'll be broadcasting from the 10th to the 11th of November. Thank you very much, everyone. Bye-bye.
Goodbye.